Hello, welcome all. I just want to do a brief explanation of our work that we've presented here in ASCO 2024 annual meeting that is based on the effect of anti-CD22 CAR-T cells secreting anti-CD19 T-cell engagers on control of leukemia progression and its comparison to a tandem CD19, CD22 CAR-T cells. Just wanted to do a brief introduction about the current state of the art in anti-tumoral T-cell redirection strategies that basically comprise chimeric antigen receptor T-cells or CAR-T cells and intravenously administered T-cell engagers that act as a bridge between the tumor cell and the T-cell. And well, they both have some advantages and disadvantages, and as you can see, CAR-T cells can actively traffic to the tumor. They have a costimulatory signal and they generate a long lifespan memory T-cell reservoir. And on the other hand, T-cell engagers can efficiently recruit bystander polyclonal T-cells and induce more typical synapses. So with the idea of picking the best from each strategy, we got to this CAR-STAB concept that comes from the addition of CAR or chimeric antigen receptor plus STAB that stands for in situ secretion of T-cell redirecting bispecific antibodies. So we've generated CAR-STAB T cells that display a chimeric antigen receptor against CD22 in their surface and secrete anti-CD19, CD3 T cell engagers. And basically, we've compared our strategy CAR-STAB to a previously validated tandem CAR directed against the same two antigens, CD19 and CD22. First of all, we designed the CAR-STAB construct by cloning two individual sequences, the CD19 T-cell engager and the CD22 CAR, separated by a 2A cleavage site, proteolytic cleavage site, into a third generation lentiviral vector. And we added GFP to monitor transduction efficiency. First, we just checked that the Jurkat cells, transduced Jurkat cells, could efficiently secrete the T-cell engager with the proper molecular weight, and that this T-cell engager could bind the target antigen CD19, both plastic immobilized by ELISA and in the surface of tumor cells by flow cytometry. After that, we just wanted to check if these CAR-STAB T cells were able to recruit bystander T cells. So basically, we set co-cultures of effector and target ratios at effector and targets at 1:1 effector target ratio, but decreasing the percentages of transduction from 80% approximately down to 10%. Basically, we measure CD69 as an early T-cell activation marker in the y-axis and GFP to monitor transduction efficacy. So, to sum up, what you can see here in the upper right chart in color blue are transduced activated T cells. They are activated because they express CD69 and they are transduced because they are GFP positive. And what you can see in the upper left chart in orange are activated non-transduced bystander T cells. They are activated because they express CD69, but they are GFP negative. So they are not transduced, but they are activated because they are recruited by the T-cell engagers secreted by CAR-STAB T cells. So you can see here that even at low percentage of 12% of CAR-STAB T cells are enough to efficiently recruit all bystander T cells in the culture and activate them against tumor cells. But on the other hand, tandem CAR-T cells were not able to recruit any bystander T cells because they don't produce, of course, T-cell engager. So after that, we wanted to see if these bystander effect was also translated into a superior cytotoxic activity. So for that, we move forward into primary T cells, and we set similar co-cultures of effector and targets at decreasing effector target radius, but adding non-transduced freshly isolated T cells to the culture, so that you can see here in orange. So basically, effector target ratios went from 1 to 1 down to 150,000, but as I said, we added non-transduced freshly isolated T cells to the culture. So as you can see, thanks to the recruitment of bystander T cells, CAR-T T cells here in green were much more effective than tandem CAR-T cells that you can see here in red. And in similar experiments in non-contacting co-culture system, we can see that only CAR-T T cells were able to kill tumor cells thanks to the production of the bispecific T cell engager that went from the upper chamber, from the transduced T cells in the upper chamber, down to non-transduced T cells in the lower chamber, so they could get activated and efficiently kill target cells here. And finally, we just perform an in vivo experiment to compare these CAR-STAB and tandem CAR-T cells with a PDX or patient-derived xenograft model of a acute lymphoblastic leukemia patient. So we injected 1 million of these PDX B-old cells, followed 3 weeks later by 3 millions of CAR-STAB or tandem CAR T cells administered intravenously. And leukemia progression and T cell expansion were followed by flow cytometry almost every week. And you can see that here at week 5, and 2 out of 6 mice treated with tandem CAR-T cells, leukemia progressed rapidly, while all the mice treated with CAR-STAB T cells showed evidence of leukemia control. And further analysis in bone marrow at week 3 revealed complete regression of leukemia in 2 out of 6 mice treated with CAR-STAB T cells, but none of the mice treated with tandem CAR or non-transduced T cells. And this superior efficacy of CAR-STAB T cells was also correlated to a higher T cell expansion in bone marrow. Just to conclude, we've generated an innovative strategy that combines 2 different and potentially complementary mechanisms against different targets, and we've called that strategy CAR-STAB. We've demonstrated that CAR-STAB T cells can efficiently recruit bystander T lymphocytes, and this enhances cytotoxic activity and helps controlling leukemia escape both in vitro and in vivo. So therefore, CAR-STAB T cells could become an alternative to single-targeted CAR-T therapies for the treatment of refractory or relapsed B-cell malignancies, especially in lymphodepleted patients with low T-cell counts. And just to finish, I'd like to acknowledge all the people from the lab in Hospital 12 de Octubre de Madrid and Spanish National Cancer Center in Madrid too. All our collaborators from different places in Spain and around the world, and thank you very much for your attention.